So how has the cell and gene therapy sort of sector, how has that changed over the past 10 years? Obviously it's growing and growing, but yeah, how has it changed in your eyes?
So in my opinion, the gene and cell therapy field came from making proof of concept in animal model 20 years ago, and then 10 years ago was the first proof of concept in the clinic. And now we see more and more commercial products. So we really see the maturity of the field going from a nice scientific proof of concept, but difficult to consider as a real product, to commercialization of products that can potentially treat a lot of patients and help a lot of patients with unmet medical need. So I think we are not yet at the level to be able to treat the number of patients, for example, with genetic diseases like sickle cell disease. But we are on the way to be able to do so. And I think it's a very interesting evolution that we saw in the last 10 years. I think another aspect is also the number of CDMOs and the number of companies in the field of gene and cell therapy that is really increasing. And this network of companies and CDMOs is really essential to be able to translate discoveries to new products.
So we've spoken about how it's changed in the last 10 years. How do you expect the sector and the cell and gene therapy products and therapeutics to change in the next 10 years? How do you think it will evolve?
I think the manufacturing will become more and more efficient and cost effective. We will be able to see off-the-shelf products with one batch able to treat many patients. We will see probably an evolution also on the type of cell line used for large-scale production. We saw transient transfixion, we saw packaging cell line. I think we are going to see more stable producer cell lines that are able to really support the development of very large-scale productions. And what we are going to see also is the evolution of economic models to be able to bring those new therapeutic modalities to patients and to have the system of reimbursement so that the patient can fully benefit of those new therapies. And even if the price could be high for now, I think that the evolution of the field will allow to have those products available for a large number of patients. And I think the last element I would like to add is that if we can find the models to make those products available to patients in Europe and in the USA and in Japan, the next step will be to be able to make them available in Africa and in different countries where, for example, sickle cell disease is a very big problem for the population. They do not access to those therapies for now, but the hope is that they can benefit of them in the long run and as soon as possible.
So we are seeing more clinical trials of gene editing products going through the clinic. Could you tell us a little bit more about that, Olivier, and the reason for that and why that trend is currently happening?
Yeah, I think it's very interesting to see the potential of gene editing. For example, we see a lot of articles on CRISPR-Cas9. So the gene editing strategies can be both used to design new models to study new drugs, but they can also be directly applicable to therapeutic strategies. So I think that there is clearly a potential to have those new technologies translate into new medicines. At the same time, we need really to assess still the safety and the efficacy in the long run of those new modalities. And I think now it's a time. Now we have already preclinical results, and we have the first clinical trial ongoing to see if those new therapeutic modalities are really meeting the level of efficacy and safety that we hope they can achieve. And I think the analysis of the results from the clinical trial will be very helpful to see how important are those new techniques. And we see that also new generation of ways to modify the genome, like the base editing or the prime editing, are coming, and that will be also interesting to compare those different generations of modification of method of modification of the genome, and to see what are the advantages and the limitation of those different generation of methods. And I think that will be very important to have these results to be able to weight what could be the impact in the long run for the new drugs coming to the market.